Yeah, I'm research group leader at INL. INL is the International Iberian Nanotechnology Laboratory, and today I'm going to talk a little bit about how we can take advantage of nanotechnologies as well as even materials on the atomic scale, such as graphene, to improve or to develop novel type of microscopies. I will not only speak about 2D material or graphene-based applications, but also look into what we can do with other type of three-dimensional nanomaterials, such as nanodiamonds, and explore quantum effects for advanced imaging. In my research group, we have three main research lines, and the research that I'm going to present today are associated to two of these research lines. One of them is to indeed develop new advanced bioimaging and sensing methodologies, and the other is to exploit quantum photonics effects or develop integrated photonic devices that have both applications in neuromorphic photonics as well as in quantum information technologies. To give some examples, an overview, in advanced bioimaging and sensing, we use nanotechnologies to, for example, develop new type of nanoscopy, near-field super-resolution technologies, nanothermometry, measuring the temperature from within the cell, or to apply nanoparticles in nanomedicine type of topics. I will not have the time today to introduce in these various applications, but I will pick one of them, the near-field super-resolution microscopy or nanoscopy, to speak a little about our recent developments where we are looking into molecular sensitivities. In the area of quantum photonics, you see here mentioned single quantum emitters in 2D materials, as well as nitrogen vacancy centers in diamond. And I'm going to present one of the applications where we functionalize nanodiamonds for a specific neurosensing approach. Last but not least, I will not have the time to talk about the third line of research, the integrated photonics area, where we have quantum integrated photonic devices on the very exploratory level, let's say, developing new technologies for areas such as neuromorphic photonic computing, with an emphasis on spiking nano-LED-enabled integrated photonic devices, a research line led by staff researcher Bruno Romeda, as well as other photonic integrated chips more dedicated to biosensing or to quantum photonics applications. Regarding the work on near-field super-resolution microscopy with molecular sensitivity, I wanted to highlight one of our recent papers where we were able to functionalize a graphene surface with a DNA linker that contains a fluorophore that is completely quenched in its starting state. And upon the addition of the target DNA, we are able to follow the DNA hybridization process by exploiting the near-field quenching in the very vicinity of the graphene that is reduced further away on the nanoscale, so that the fluorescent dye that has been completely quenched in the starting of the experiment is unquenched, and we can detect its fluorescence intensity increasing over time. And the measurements, how they look like, are explained here. We take a recording of wide field fluorescent intensity images, and from this recording we can see individual spots that are each of them changed over time. So if we follow the intensity spots, and for each of these locations where we have fluorescence at the end of our experiment, we can extract curves where we have first a background level, then the intensity that is due to the background when we performed the pipetting of our target DNA, third, a waiting time until an intensity increase is happening, and fifth, a maximum intensity that is stabilized after a certain time. We can associate these intensity levels to different unfolding statuses of these DNA hybrids, where we have here a fully folded and standing fluorescence DNA hybrid. This was not enough to us to just follow the molecular dynamics, but indeed we contacted our collaborator, Pedro Ipuim, a research group leader at INL as well, who is an expert in graphene, with whom we did this work. But we asked him to further develop the graphene into a graphene device where we can perform some electrical gating. Now we would like to move our DNA molecules and not only follow their natural kinetics, but to gate the device and to move in a deterministic manner on molecular level the fluorophore up and down. And this is work performed at the time by master student João Acevedo, who is now a PhD candidate, who added DNA origami structures to our graphene substrate and now could follow the motions on demand after a gating of the graphene device. And so the device looked schematically a little bit like this. We have a tetrahedron constructed of DNA, and then we have at different height positions attached a fluorescent label, an A248 molecule, and we have the graphene device, which can be gated by a voltage attached to the device. What we can do now is to move the position of our fluorophore from a highly quenched to a less quenched level, which changes the fluorescence lifetime of our dye. And you can read these in more details in this paper mentioned here, in Advanced Materials Interfaces. So what we do now is to look at the interaction that we can have of a fluorophore with the dye in vicinity of graphene. And you can see the distance-dependent quenching and the theoretical expected curve of the fluorescence lifetime measurement here. And we perform experiments first with the test level where we are adding our fluorescent marker at different locations in our DNA tetrahedron origami structure and deterministically. And we work now with DNA origamis labeled with a dye, for example, located at 12 nanometers away from the graphene or at 17 nanometers away from the graphene. And every time we can follow the fluorescence lifetime performance. Before gating the device, we test that our origamis are indeed following the theoretical curve, which you can see here on the bottom right, where we have now our fluorophores attached at these different locations, such as, for example, at a distance of 20 nanometers. And we can see that a 20 nanometer distance is associated to a lifetime measurement of 2 nanoseconds, simply using an inverted microscope, a fluorescence lifetime imaging microscope, where we have a picosecond laser coupled into an inverted microscope, and we detect the photon arrival times for each of these photons, having the ability to measure for each of the pixels in the image the fluorescence lifetime information. As a test, we see that our DNA origamis are indeed intact on top of the graphene using AFM characterization. If we now add our gating to these graphene devices, we can see that we have a typical intensity or also typical lifetime for a fluorescent dye. But if we change the voltage on the gate, we can move the intensity down, and once removing the voltage again, we go to the starting position. So the intensity can be tuned, but we can also see that with increased voltage, the fluorescence lifetime can be changed gradually. We can analyze the effect by the shift of the Fermi level in our graphene device, and therefore have developed a very interesting nanomechanical device that could be used to sense distances. Also, once we have, for example, an interaction with these tetrahedrons and other biomarkers or linkers. So one could imagine that the distance of our labeling dye could be used as a way to measure the height of the entire tetrahedron itself, which may depend on electronegativity or a complex binding mechanism on top of the origami surface. But as I mentioned in the introduction, we would like to move away from only 2D carbon, but look into what we can do if we have 3D carbons. And especially if we work with diamonds, we can think of bright, shining nanodiamonds that contain fluorescent NV centers, and we can use them in future, hopefully, to measure neurosignaling processes. What we need to do is to bring our nanodiamond sensors in very close proximity of axons in neuronal cells. And we do this by adding a functionalization to our nanodiamonds. In this case, via a biotin streptolivine linker, we are moving our nanodiamonds directly to antibody called CAV2.2, which is a channel protein that is quite present in different neurocellular systems. We can now aim to perform optically detected magnetic resonance experiments with such nanodiamonds that are in very close proximity to the neurons where the action potential, a change of charge, is happening. If this change of charge, which are traveling charges along the neuron, occurs, we expect a magnetic field that is following the neuron. And this is the magnetic field that we would like to pick up through an optical interrogation. But before doing that, we need to make sure that our nanodiamonds indeed carry the intended functionalization that we aim to have. So we test whether we could bind our antibody to our particles. Indeed, our particle size slightly increases upon the functionalization. The zeta potential is still negative enough that they are separated, and they do not change the optical properties, which is great because we want to explore the optical properties for the neuronal sensing later on. We can show that the secondary antibody indeed is labeled with an test dye and overlapping with the nitrogen vacancy emission of the nanodiamonds themselves. We successfully test that these nanodiamonds with their functionalization are not damaging to any of the neuronal cell samples that we have. We worked with a cell line called SHSY5Y, as well as with dopaminergic neurons and hippocampal neurons from mouse models. And we can see that indeed they are biocompatible. They do not disturb the axon growth. So we can see extended axons that are looking very similar between treated cells that were treated with nanodiamonds or with functionalized nanodiamonds. Of course, another control has been made without any diamonds. And also the neuronal excitability is not modified, as can be seen with this calcium ion imaging, where the neuronal signaling is intact. We can show that the targeting, once the nanodiamonds are functionalized with the antibody, is indeed increased. We have an increased factor of 2 to 2.5 to reach the locations of these nanodiamonds in the positions where we can measure action potentials. And we demonstrate that indeed the optical detected magnetic resonance sensitivity of NV centers in the presence of these cellular models can be observed by a simple test experiment where we expose our neuronal cells to a magnet or not. In the presence of the magnet, you can nicely see the splitting of the ODMR traces and could then extract magnetic information from these signals. We have to mention that at this moment, the sensitivity for magnetic field sensing is far from the required sensitivity that we would need to record magnetic fields created by neurons. But for the temperature sensitivity, we reach very relevant ranges that could be interesting, especially in the area of cancer research. We also think that we can increase the sensitivity if we move to pulsed ODMR protocols. What you see here are continuous wave ODMR experiments, which have less sensitivity. With that, I would like to stop and conclude that first thank the students and the researchers involved in the experiments that I showed, and also thank the funding agencies specifically Lacaitian FCT for a project called Diamond for Brain, as well as national funding via two projects, the Quando and the Diamond Connect project, and some cost action project. And you can see here already in the banner that I would like to introduce an event where we can discuss advances in bioimaging technologies in much more detail. And I invite all the EPIC members and non-EPIC members, of course, everyone that is interested in microscopy, to join us in November, from the 5th to the 7th of November in Braga, Portugal, where we will be co-organizing together with two other institutions, the INL, the ICVS, the Medical School of the University of Braga, as well as the 3Bs, this exciting meeting, which is called the Spanish and Portuguese Advanced Optical Microscopy Meeting. Maybe some of you know this event. It is, even though it is Iberian, we have international speakers, invited speakers, and international guests. And we typically have, besides the talks, also technology workshops where microscopy companies have the chance to host technical workshops. So as well as booths as at an expo. So I invite you all to participate and to contact me for more information and opportunities if it is having an exposition, sponsoring the event, or participating to learn some of the new trends. Some of the new trends, we will have focus sessions in technologies for 3D models, health and disease model imaging, about AI and microscopy, about new tools for neurosciences, as well as quantum imaging and sensing sessions. And then, of course, the hot topics in microscopy applications and methods, and hot topics in AI and data analysis. And with this, I would like to thank you all, and thank you for the opportunity to share our news here at this EPIC meeting.